In this video, we're going to talk about using studies wisely. Often, in statistics, we create experiments, for example, around sample populations of a larger population to infer things about that larger population without either experimenting on or sending out surveys to the entire population. So, something that happens with that is what we call sampling variability. And that refers to the fact that different random samples of the same size of that bigger population produce different estimates. Sometimes those estimates are close enough to each other, but sometimes they're really far apart. But that should make sense, especially if your experiment is on humans. So let's say, for example, I got a really big bag of beads and I want to pull a number of beads out of that bag and calculate the percent that are red. Now I happen to know that in that giant entire population of beads in that bag, 30% are red. So if I conduct this experiment and I pull out 20 beads and I create a dot plot of the percent of those groups of 20 that are red beads, I get this example of a dot plot. It's pretty spread out. Notice that the range is from 10% red to 50% red. Now, what if I decided to pull out 100 beads at a time instead of 20? My dot plot would look something more like this. So that's something that we get into called sample size. And we know, and I bet you even knew before you took this class, that sample size is important. The larger the sample size, the more accurate or the more trustworthy your results. So larger samples tend to produce estimates that are closer to the true population value than smaller random samples. Estimates from larger samples are more precise. When you are designing an experiment, you want to make sure that you allow for inferences that could explain cause and effect. But we should only conclude changes in our explanatory variable that cause changes in the response variable if the results are what we call statistically significant. So when the observed results of a study or an experiment are too unusual to be explained by chance alone, the results are called statistically significant. Remember back to the caffeine and pulse rate experiment mentioned earlier in this chapter. Let's dig that one back up. So let's say that a stats class had 20 student volunteers and they were going to conduct this experiment. Does caffeinated soda have an effect on pulse rate? So 10 random students were assigned to drink caffeinated soda and the other 10 non-caffeinated soda. And then they measured the pulse rates in beats per minute before and after consuming the soda. So what we have here is a chart with the data of the change in pulse rate. So they took the final and subtracted it from the initial pulse rate. And so what you notice here is that we've got caffeine, and these are the differences in their pulse rate. It ranges from no change, so 0, up to 8 beats per minute more. So that means that the average change, if you wanted to calculate that, is about 3.2 beats per minute for the caffeine group. Now, you could do the same with the caffeine-free soda group, and it looks like their pulse rate it ranged from slowing down by 2 beats per minute to speeding up to about 5 beats per minute. And if you average those out, we get an average change of positive 2 beats per minute. So it looks like, at least from this data, the caffeinated group, the caffeine soda consumers, their average change in pulse rate in beats per minute is about 1.2 beats per minute more than the non-caffeinated group. So how do we explain that? Well, there are two major ways that we can explain that, at least for this experiment. The first is that caffeine doesn't have an effect on pulse rates, and that difference, that 1.2 more for caffeinated soda consumption, is that it happened because of chance variation and random assignment. So maybe if we shuffled some students around in each group, there wouldn't be a 1.2 beats per minute pulse change, perhaps. Or, hey, maybe caffeine does increase pulse rate. So essentially what we're trying to figure out is that difference of 1.2 beats per minute a statistically significant amount or not? In order to better answer that question, the class decided to do a little trial. And it's not so little. It actually probably took a lot of time. But anyway, they distributed the 20 changes in pulse rates. So all the things that I've highlighted in green here. So what they did, they used cards actually, is they put all of these numbers, so 8, 3, 5, etc., on each, an individual card each. And so they shuffled them up and they assigned 10 to be the caffeine group and 10 to be the non-caffeine group. So that could mean that 8 stays in caffeine or it switches to no caffeine. Essentially, they just swapped around the change in pulse rate because they wanted to see if, hey, maybe it was random distribution that caused this change and not actual like caffeine has an effect on pulse rate. So you swap them around. They did this 100 times. So what they did each time is they calculated the mean change in their new caffeine group and the mean change in their no caffeine group and found the difference from subtracting no caffeine from caffeine, just like how we found that 1.2 difference. So they did that 100 times. Again, you don't need to use cards. You could use technology to help you with that, but they use cards. So this is the dot plot that result did. So what we're looking at is the numbers from -3 to +3. So that's going to be the simulated difference, the caffeine minus the no caffeine, just like how we got the 1.2, remember? In mean change in pulse rate. So what ended up happening? Did we get numbers close to 1.2 or not? We got a pretty big range. So it went from -3 beats per minute's change to +3 beats per minute's change. And how we compare that to 1.2, first of all, we have to locate 1.2. And in 100 trials, 19 times, so 19%, the difference was 1.2 beats per minute or greater. So what does that mean? That means that getting a difference of 1.2 really isn't that unusual. In 19% of the trials, they obtained a difference of 1.2 or greater simply due to chance variation. They really varied this up. They really did. So I trust this data. Mostly because I don't want to replicate it myself. So therefore, the results of the original experiment are not statistically significant. So we cannot conclude that caffeine does, in fact, change or increase pulse rate. Damn. So the overarching theme of this video is something that we call the scope of inference. We've been talking about inference, its wide variety of uses. The scope of inference has two main points, which we've also touched on in this video and in others, but let's summarize. We love a good summary. So random selection of individuals allows inference about the population from which the individuals were chosen. So that's that sample population making inferences about the larger population in which you're doing an experiment or study to collect data on. And then the second part of the scope of inference is that random assignment of those individuals to groups allows inference about cause and effect. And of course, I'd put a little, asterisks in there and say, make sure you've got some statistically significant data to back up that cause and effect. Now, I love a good chart, so let's summarize the scope of inference into this chart based on these two ideas. The first being, were individuals randomly selected? And that's either a yes or a no. And then you compare it to, were individuals randomly assigned to groups? Yes or no. So if you have yes, individuals were randomly selected, and also yes, that they were randomly assigned to groups, you can say that you can make an inference about the population. Yes. And can you make an inference about cause and effect? Yes. So double yes means two yeses here. Now, a yes for were individuals randomly selected, but no, they weren't randomly assigned to groups. You can make an inference about the population, but you can't make inferences about cause and effect. So if you have individuals that were not randomly selected, but they were randomly assigned to groups, you can't make an inference about the population, but you can make an inference about cause and effect. And you probably guessed, if you have a double no, they weren't randomly selected, no, they weren't randomly assigned to groups, sorry, no inferences about the population and no inferences about cause and effect can be drawn. So we know that a well-designed experiment can showcase cause and effect. And when it's done well, that's a beautiful thing. But sometimes it's just not practical or it's not ethical to conduct an experiment to look for cause and effect. For example, does smoking cigarettes cause lung cancer? Now, I know there's been a lot of experiments on that, but I'm not going to create an experiment and have people smoke, because that to me is unethical. So there are several criteria for establishing causation when we can't perform an experiment. First up, the association between the two variables, the explanatory and the response, it is strong. Using the smoking and lung cancer example, oh, definitely, that association, very strong. The second is that the association is consistent. So there have been many studies and many experiments over many years that has consistently said that lung cancer and smoking are related. Third criteria is that larger values of the explanatory variable are associated with stronger responses. In our example, it's been proven that people who smoke more cigarettes per day for longer periods of time have a higher cancer risk and get lung cancer more often. And those people who stop smoking reduce their risk. Fourth criteria, the alleged cause precedes the effect in time. And essentially that means that the cause has to happen before the effect, which that's how time works. That's how cause and effect works. But in our example with lung cancer and smoking cigarettes, there's usually about a 30-year lag in when a person starts smoking and when they're diagnosed with lung cancer. Now again, it's not always 30 years, but obviously you start smoking first, that's your cause. Your effect later is that you are diagnosed with lung cancer. Cause and effect. And then the last one is that the alleged cause is probable. So in our example with lung cancer, experiments with animals have shown that tars from cigarette smoke do cause cancer in those animals, which is sad for everybody involved. One last thing that I want to drop in this video before we end is something that we call data ethics. So that is when we consider, is this data collection, is this experiment that is going to lead to data collection, or this observational study that's going to lead to data collection, is its methods, is its hypothesis, is it ethical? Now, you've been a human long enough, once again. That is a very hard question to answer, and oftentimes the answer is not a straight up yes or no. Sometimes it's very clear, like that is unethical, but oftentimes it's a huge debate among scientists and researchers and data analysts. So here are 3 different examples of this might be a little blurry ethically, maybe it's not for you, but give a think to each of these 3. So the first one, a promising new drug has been developed for treating cancer in humans, but before giving the drug to human subjects, researchers want to administer the drug to animals to see if there are any potential serious side effects. Ethical? Unethical? You decide. Second, are companies discriminating against some individuals in the hiring process? To find out, researchers prepare several equivalent resumes for fictitious job applicants, with the only difference being the gender of the applicant. They send those fake resumes to companies advertising positions and keep track of the number of males and females who are contacted for interviews. Ethical? Unethical? Somewhere in between? Last up, will people try to stop someone from driving drunk? A television news program hires an actor to play a drunk driver and uses a hidden camera to record the behavior of individuals who encounter the driver. Ethical? Unethical? You decide. So do not fret. There are rules and laws, at least in this country, in place to make sure that experiments and studies that are run, especially on people, are as ethical as they humanly can be. So 3 main things to understand about data ethics in the larger context, or in the especially the professional context. First, that all planned studies must be reviewed in advance by an institutional review board charged with protecting the safety and well-being of the subjects, especially if they're human or animal. A lot of universities will have this kind of review board because a lot of research is done at the university level, and they are obviously there to make sure that the rights and welfare of any of the subjects are protected and maintained. Second, all those individuals who are subjects in a study must give their informed consent before data are collected. Now, That's a lot of different things, but that is a lot of paperwork. And in order to be experimented on, at least, a human has to give informed consent in writing. And lastly, all individual data must be kept confidential. Only statistical summaries for groups of subjects may be made public. So there's no identifying information that is released anywhere about an individual or a group, because that just breaks all sorts of confidentiality laws. Yeah. All right, that's going to do it for this video. Thanks for watching.